All right. Good afternoon, everyone. Thank you for coming to this session on animal health and well-being. My name is Paul Fricke. I'm a professor in the Animal and Dairy Sciences Department here at UW-Madison. I'm also on the Dairy Innovation Hub Steering Committee for UW-Madison. We have, in this session, two longer talks to begin with, two 25-minute talks, followed by four 12-minute talks. So I'm looking forward to some good talks and good discussion. It's my pleasure to introduce my cohort in crime up here, who's going to MC with me, Dr. Jimena Laporta. She's an assistant professor of animal and dairy sciences at UW-Madison. Her research investigates how autocrine, systemic, and environmental factors affect the regulation of mammary gland development and function. Additionally, she researches how these factors affect milk synthesis and composition. And Jimena is going to be talking to us about active ventilation for dairy calves, a step toward improving health and welfare during hot summers. Help me welcome Dr. Laporta to the platform.
All right. Can you hear me okay? Yes. Well, thank you, Paul, for that introduction, and thank you, everyone, for being here today. What a great way to start this afternoon session than talking about heat stress, right? It's finally getting warmer here. I think so. It didn't look like that this morning, but we're getting there, slowly but surely. So when we think about heat stress, and this is already not working. Let's see. It's on. Oh, there we go. Maybe. Oops. It's on.
Yeah, it is on.
I can.
I had it working before.
Yeah, me too.
There,
anyway,
let's,
I can.
work on it.
Oh, it's not. It's because
Gotta
it's
put
not.
it in presentation mode.
Yeah, there. We thought we had all this figured out, but
Try it now.
there we go. All right, thank you.
I think we got an extension guy up here.
Good thing we practice. So when we think about heat stress, we typically think about our lactating cow and how to prevent the drop in milk production and intake that they suffer when they are under heat stress. But heat stress profoundly affects dairy calves as well, and this effect starts even before they are born, okay, during this prenatal stage or in utero stage. And our lab has done research to try and understand how in utero heat stress affects many organs and systems, including gastrointestinal function and the ability of that calf to absorb immunoglobulins, the ability of that calf to thermoregulate, immune function, the impact on immune function, metabolism in general, the ability of that calf to respond to stress later in life. But most importantly, we have done a lot of work understanding how in utero or prenatal heat stress derails mammary gland growth, the trajectory of that growth, and how that has long-term effects or carryover effects for multiple lactations, multiple years, and actually multiple generations. So we're not going to talk about this today. Actually, today we're going to talk about the effect of heat stress in the postnatal calf. Once that calf is born, during the pre-weaning period that typically is 8 weeks. And we're going to talk, this is an area that we know a lot less about. It's a fairly new research area in my lab for the past 3-4 years now. And we do need to learn a lot more about that. So we're going to focus on this today. All right, so when we think about heat stress in dairy calves, there's a general misconception that calves, because they are more thermotolerant than mature cows, that they don't need to, they don't feel the heat, they don't need heat abatement. Because they have a narrower thermoneutral zone than mature cows, they have a larger surface area to mass ratio to be able to dissipate heat. Obviously, they are not lactating. They have a fairly undeveloped rumen, so they don't generate that much metabolic heat compared to mature cows, lactating cows. So they're not affected by heat stress. But in fact, that's not true. As you can see in this video, this is at the Arlington Dairy Research Unit a couple of summers ago. When calves are exposed to high temperatures and humidity, they are going to be, in fact, heat stress. As you can see here, that manifests in increased thermal indices, including respiration frequency. You can see how hard that calf is breathing, increased respiration rate, reduced intake, poor health, et cetera, et cetera. Now, the problem is that daily on-farm monitoring of these parameters is challenging. We have a lot to do in the farm, and it's time-consuming. But I think despite the fact that we don't see an immediate effect in production like we do in mature cows, it is an important welfare concern that we need to address, especially in the face of climate change. Summers are getting longer and hotter, so there's a lot more calves, especially here in Wisconsin, that are going to be under the influence of heat stress. So it's an important concern despite not having a tangible, immediate response, okay, like we do in dairy cows. And also the fact that I'm going to show you today that there are long-term effects of postnatal heat stress later in life. All right, so when we think of developing strategies for heat abatement in dairy calves, we have to think about housing type. We all know that there are many ways we can house calves on a dairy farm. Most dairy calves in the US are housed individually in hutches, and of those, more than 40% are outdoors, exposed to solar radiation, and we know the greenhouse effect that can have. It gets really, really warm inside that hutch. So luckily, we have best management practices that are widely implemented on farm that we use to avoid that overheating, and that includes different hutch materials, supplementation of extra shade, and also ways to improve passive ventilation, natural ventilation. I don't know if Jennifer is here, but she's there. Hi. She has done a lot of research on this, and this is a great way to promote heat abatement through the addition of windows and rear vents as well, changing the hutch orientation and also hutch elevation. These are great ways that we have to avoid overheating, improving natural ventilation. Now, there's also increasing in popularity for group housing, whether it's in pairs or in larger groups. And so when we think about calf barn ventilation, the emphasis has been in air exchange per hour. I think roughly, correct me if I'm wrong, it's 40 exchanges per hour what we're targeting, but that's mostly to maintain a fairly good air quality and not so much for cooling purposes. So despite the type of housing we're talking about here, the implementation of active mechanical cooling for dairy calves, it's very limited to a handful of studies, including these and the 2 or 3 I'm going to show you today. So I think there's more research that needs to be done in this area. So we have a problem. Calves do feel the heat as well. And in order to find solutions, we started asking some questions. These are some of those basic questions. Can we actively cool calves? How do we do it? Indoors, outdoors, individually housed, group housed? Are there any short-term, long-term benefits of doing so? Do we get a similar response in different environments and things like that? So to do that, we conducted a series of studies, one at the University of Florida before I moved here, with group house calves, and that was funded by the Milk Check-Off. This is a milk co-op there. And 2 studies here in Wisconsin with outdoor hutch calves funded by the Dairy Innovation Hub. So thank you. So this first study was done in Florida, and the objective was to provide active ventilation to group house calves. That's the way they house their calves there. In summer, undeniably a hot summer, as you can see here. This is a picture of what the design of those pens were. We have 8 pens, automatic feeders. We have an open side barn with mobile curtains. And we added active ventilation via fans. We had one barrel fan here. It's a 42-inch in diameter. And we target it so that the airspeed was at the calf height level. And then we have this oscillated fan above the ground. These were set on a thermostat at 21 degrees Celsius or 70 Fahrenheit. That didn't matter because they were on 24/7, given the ambient temperature that you see right there. So the calf height airspeed in those pens that had fans was on average 2.5 meters per second for the active ventilation versus 0.15 for the passive ventilation. You can see in this graph, this is airspeed, and this is the pen location. We assess different locations of that pen. And in gray, you have the fan pens, and in red, you have the natural ventilated, no fan pens. So you can see that in all locations, it's higher as expected. We had no idea where to start, so this is where we start. And as a reference here, we have cow side target airspeeds between 1 and 2.4 meters per second are effective in promoting thermoregulation in mature cows. So we were kind of right on target, but we certainly didn't know what to expect. But we were we were right in those airspeeds because forcing that air movement across the pen triggered thermoregulatory mechanisms in the calf to cool off. And this is depicted in these two graphs here where we have respiration rates, so the breaths per minute of those calves, and this is rectal temperature, and we measure that at different times of the day to capture the diurnal variation. So we see here 7 a.m., 1 p.m., and 7 p.m. And Brittany's here. She was part of that study, so I'm sure she remembers the 7 a.m. Or the 7 p.m. that was hotter for sure. But what we can see here is that the fans that are represented in these gray bars at all times of the day, those calves had lower respiration rates and lower rectal temperature. So the fans were effective in inducing a thermoregulatory response in those animals. More importantly, they improved milk and grain intake. We see here milk intake, grain intake in the y-axis across postnatal weeks from birth to weaning. In gray, we have the fans, the pen with the calves with access to fans. And we can see that on average, those calves drank 1 liter more of milk per day. And also on weeks 7 and 8 in particular, they ate half a kilo more of grain. So that we know that how important that is for rumen development. Very interestingly, we see that provision of fans not only improved grain and feed intake, but it also changed the feeding patterns. And this is very important at a farm level. Those calves that had access to fans drank 0.5 liters more per visit. So they not only visit more times, and they weren't rewarded because you have to set up a limit of what they can eat and when. They were insisting, they wanted more, and every time they went, they ate more than those that were heat stressed without any fans. And also, all calves decrease intake at the hottest time of the day. I would say that was between 11 and 5 p.m. They all decrease the intake, so they eat more in the morning and later in the evening. But the provision of fans kind of rescue that, and those calves that had access to fan ate more during those hottest time of the day. So I think this is very important finding. But most importantly, and you will like this, provision of fan improved reproductive outcomes. We followed these animals for 2 years. And what we found, this is preliminary data, but what we found was that those that had access to fans, remember those are in the gray lines, got pregnant sooner than the ones that didn't have access to fan. And when they start their lactation 2 years, even more later, they produce more milk. On average, 2.4 liters more milk the first 30 weeks of lactation. So pretty impressive, and this highlights the importance of this early life programming that we talk about, perinatal programming, and what we do management-wise early in life, how it really can have carryover effects for years down the road. What we do here during pre-weaning period really matters, and you can see how it impacts first pregnancy and later on lactation. So that was great. This is very exciting. So I move here and I start seeing a lot of hutches. So what do we do now? How do we do this in this setting, in this type of housing? So we had to get a little bit creative, and I'm glad that the Dairy Innovation have liked that idea. And so we were Our goal was to effectively provide active ventilation now in outdoor hutches. And we wanted to evaluate the microclimate inside the hutch and also the physiological responses of the calf. So put together a team here. We did a Latin square pilot study prototype. No idea where to start, but we had to do it. So we started, and this is what we did. We compared these 3 treatments: passive, natural ventilation. We did this in collaboration with Dr. Doria. I don't know if he's here or not. I saw him earlier. And Dr. Jennifer Van Os. We did passive natural ventilation with the rear upper window open, and the lower vents where we can't see it here, but they were 50% open. Minimal ventilation with all the windows closed, and mechanical ventilation. And for this, we turned to solar power fans. And what we did is we redirected that air using this tubing attached to the upper window. And we did set a thermostat here for 21 degrees Celsius. Different from Florida, the fans were mostly off at night. Okay, and this is a very important consideration when we think about heat stress in the Midwest and in the Southeast as well. So they were off mostly at night. And what we found briefly is that those fans were on 84% of the time, mostly off at nighttime. And these are the air speeds that we achieve inside the hutch. You can see how that's measured in this video. We have 1.74 meters per second for the active ventilated hutches, 0.21 for passively ventilated hutches. I would say this is the industry standard, and 0.5 for minimally ventilated hutches. All the hutches got warmer inside at the hottest time of the day. With the naturally and actively ventilated hutches, we were able to decrease that internal temperature compared to the external temperature, but we didn't see any difference between the natural and the actively ventilated hutches. This was a Latin square. The exposure was 4 days. So we didn't capture any differences in air quality in this short time. Now, to determine the impact of this type of ventilation, these 3 treatments in thermal responses, we had to apply a restriction because calves are free to go in and out as much as they want. And so we had to make sure that they stay inside so we could measure these things to evaluate how well these thermoregulatory responses were responded. So what we did was a 30-minute restriction outside. We measure all the thermal indices. Then we lock them inside 30 minutes. We measure this again, and then we calculated the difference. What I'm showing here is the positive effects on thermoregulation for respiration rate. So here we have the difference in those 30 minutes out, 30 minutes in respiration rate for active, passive, and minimal ventilation. And you can see that there's a linear decrease in respiration rates depending upon the ventilation type. So respiration rate decreased in all groups as expected because they're protected from solar radiation, but active ventilation decreased that even further. Now, like I said, calves are free to go in and out voluntarily. So we wanted to assess, are they using it? Do they like it? We didn't know. So we assess these via cameras, the calf position and location within the hutch, using cameras and computer vision systems. So we could detect if they were outside lying, inside, or outside standing. Not going to go over into much detail here, but I'm going to tell you that all calves spend less time inside the hutch between 12 and 5. It does get hot inside no matter what. But this was surprising. We were expecting that putting some fans is going to be cooler, they're going to like it better, they're going to use it. But we did find the opposite. They actually, this was a few minutes difference. I think it was like 15, 20 minutes. Is it biologically relevant? We don't know, but it was significant. So we had to talk about it. So they spend slightly less time inside the hutch. So some of the things that we thought potentially there was too much air blowing in their face. It was not comfortable for them, 2 meters per second. Elevated noise, that fan generated some noise. It was 20 decibels higher than the other ones. Or maybe the short nature of the study. They need more time to acclimate, to see what's going on, and then started using. So you live and you learn. So we took that information and we proceed to do another study here with some modifications that were implemented to reduce the airspeed and to extend the length of exposure. So now this was longitudinal study, 32 calves from birth to weaning. The fans were on for 4 weeks, the first 4 weeks of life, and they were off then until weaning. So we target the 4 weeks, first 4 weeks of life. We add a silencer here to reduce the noise, and we bifurcated these two to decrease the speed inside the hutch. So we did these changes, and what we found now is that we have 1 meter per second inside that active ventilated hutch versus 0.07 meters per second in the natural ventilated. We're comparing two treatments now. And positive effects in thermoregulation. We extended the restriction time now to 1 hour outside, 1 hour inside to give them more time to thermoregulate. And we see that all of them decrease skin temperature, rectal temperature, but the active ventilation calves further decreased those parameters. So it was very positive for them. Have the numbers here. Now, I think the most important results of this project was the improvement in hatch microclimate. The hatch that had fans had fewer colony-forming units. I have to thank the vet school here for lending me this machine. We collect air samples and we plate them in agar plates for 35 degrees for 24 hours. And a picture can tell you more than 1,000 words. So I don't know if you can see here, but there were less colony-forming units in those hutches with fans compared to non-ventilated hutches. We also have reduced ammonia levels. We use these handheld devices to be able to measure in real time what the ammonia levels were, and they were decreased as well. And this is my last slide, but we were a little worried that this could be detrimental for their health. They are going in with 2 days of life, air blowing in their face, pneumonia. That's the first thing we thought. So we collaborated with Dr. Olivet from the vet school, and we did a bunch of lung ultrasounds. And we found that overall calves were healthy. There were only 3 calves that were diagnosed with clinical pneumonia, and there was no treatment effect on ultrasonography subclinical pneumonia. So we were good there. So with this, we're starting to see more and more of these as we drive through Wisconsin farms, and just want to bring that up. It seems that active ventilation has a promising future, I would say. There's a lot more work to do and assess and test, but think that solar panels can provide shade for cattle and also could be a potential sustainable energy source to cool cattle. So with that, I'd like to thank my collaborators, the Arlington Dairy staff, funding, and this amazing group of students that have done all the work that I presented here today, particularly Bethany Day-Sen, Grace Larson, and Emily Tabor that are not here, but they did most of this work. So, but to all of them as well, because they all collaborated. So thank you, and I can take any questions.
All right, the floor is open for questions. I'm going to be Phil Donahue, for those of you that remember Phil Donahue.
Good job, Paul. Get your steps.
So first of all, I'm not an animal person. I'm an engineer by trade. But just wondering for your design, so you have on one end, you have the fan blowing air. So for sure, the heat stress will be a number one factor. But I'm thinking about the other factors involved in this study, like the behavioral changes due to noise, due to the vibration of the fan, the air speed, air blowing in the pan. How those play in the picture? Like, are those accounts for 10% or they do not affect anything? Like, what's your take on that? Thank you.
Thank you for the question. We try to account for those things, and we measure the noise level because we knew that that was going to be a factor. It was a little noisier inside that hutch. But I was surprised how high the normal levels are on a farm, if you think about it, right? So this was 20 decibels higher compared to the controls. So we try to account for all of those factors. I don't know what's the percentage that they play in all of these, but we try to be aware of those and measure them as best as we can. And I have in mind more studies that, every year to improve a little longer exposure. I think it will be best to have those fans for the entire pre-weaning period. I didn't show here, but we also measure body weight and a bunch of other things. We don't see any differences during the treatment application during those 4 weeks, but when you turn them off, those 4 weeks up to calving, the ones that had the fans, they had a little bump in growth. They are like, what are you doing? It was nice here, and now you're taking it away. So I think that we have other ideas on how to improve these even further. But yeah, I don't know if that answered your question, but.
Okay, we got time for a few more questions.
So regarding the fans, I don't know if you clarified, did you blow air into the hutch from the rear, or did you pull through from the front and out?
Inside the hutch.
Do you think it would make a difference if it was blowing on them versus being pulled through on an impact on an animal welfare perspective?
I don't know. I think we have to do that project. That question came up when we were designing all of these, and that's a fair question, and I don't know the answer for it. I think we have to request more funding to do that.
Yeah, that's always the answer, more funding. Other questions? Yeah. I know, shameless. Oh, Mike Peters back here. Come in here, Mike.
Great talk.
Thank you.
Did you see any effect on flies in the hutch with having the airspeed in the hutch? And do you think that affected your CFU counts you presented?
Thank you, Mike, for that question. Not really. There weren't that many flies. I think it was pretty good management. Thank you, guys. Calves were super healthy. Like when we were analyzing this data with Terry, we were looking at IgG, and because we have to enroll them on a rolling basis, we couldn't select, like only calves that have good immunoglobulin. So we had a good range, but they were all super healthy. There were not that many flies, but even there, we can pick up that difference in colony-forming units. So I think it's important. I think this discussion came up during Grace is one of my students that did this project during her committee meeting. We want to repeat this in an average managed farm, because this was very good. So.
Okay, we got one last question here. We got one more.
For the study you did in Florida, you measure consumption, like grain and milk. But I'm not sure if this automatic system, do they have access to water? And did you measure water consumption?
They do have access to water, but we didn't measure. It was like a large water trough, and we didn't, no. But we could measure grain, which was great, and that was incorporated into the system. So it was a scale that every time they ate, it was measuring. So that was really great.
Okay, why don't you? We'll
yes,
get
sir.
Hilario
well, thank
wired
you.
up. Yeah, let's give her another hand. All right, so next up, we've got our second 25-minute talk, and I'm going to go ahead and turn the mic over to Jimena.
You have to turn it.
Should be.
Yeah.
Oh, that thing fall off.
Yeah, you have to turn it. Oh, yeah.
Sorry.
Just.
Is it working?
I think it's working.
All right. Well, I'm going to turn it to our next speaker. This is another 25-minute talk, like Paul said. And this is Dr. Hilario Mantovani, and it is Mantovani. It's a great example of the talent that we're hiring through the Dana Innovation Hub. Dr. Hilario Mantovani, he is an assistant professor in the Animal and Dairy Science Department at UW-Madison. His position is funded by the Dairy Innovation Hub, and his research focuses on the function and ecological interactions between anaerobic microbes to colonize the gastrointestinal tracts of ruminants. So help me welcome Hilario.
Okay, thank you, Dr. Laporta, for the introduction and for the Innovation Hub to be here today to present some of our results. The title of the presentation gives away the outline of talking today here. So we have been interested in therapies to control infectious disease in livestock, with a particular emphasis in ruminants. And today we will be focusing particularly on bovine mastitis as one of our models of infectious diseases that we wanted to develop alternatives to. So this is a major topic that has been on the news for quite a while. It's not something new, the use of antibiotics in animal production in general. And this became even more important in the early 2000s when antibiotics started to be banned in terms of use in livestock production. And this started to happen like in 2006 when the European Union decided to ban the use of prophylactic use of some certain antibiotics based on studies that were developed initially in Sweden and Norway. And even more recently, we still are seeing this on the news, and some regulations are being imposed on the use of these antimicrobials in animal production as well. So this was February last year when we had the European Union again banning the routine use of antibiotics on farm animals, some also for prophylactic use. In the United States, next month in June, we will have the use of antibiotics for livestock being prescription only. So this starts like next month. So there are always these issues about the use of antibiotics in animal production and a general perception of the public. And like this morning had like a great talk about that, how the public perceives the use of certain practices in animal production systems, right? So, and antibiotics is one of them. If you enter like into a grocery store, you'll see like products, I'm not trying to make propaganda to any of these brands, but you see a lot of these things related to meat consumption or milk products. They're free of antibiotics. So the general idea of the public is that antimicrobials or these antibiotics that are used in animal production are kind of bad for the general public. So and of course the food industry tries to adapt to that and develop products that are free of these compounds as well. So these kinds of raises a problem because if you're talking about agriculture intensification and increase of world population, we have like more and more animals put together, like in the same environment, in like smaller distance, and that is a major problem for us when we think about the general operation of this practice. So other things that tie with this general perception is the fact that antibiotics are heavily used in animal production. So in the United States, twice as many antibiotics are used in animal production compared to human use. And this is not an issue only in the United States. Of course, in Europe, it's also a problem, and the Americas the same. And the critical aspect is most antibiotics that are used in animals are also medically important. So if you look at the classes of antibiotics that have like are considered to be medically important, you see like a whole bunch of antibiotics that are also used in the clinical practice. So this kind of like imposes a crossover of this resistance. And if you think about the concept of One Health, where the health of the animal is integrated with what happens in the environment, in terms of environmental health, and also human health, this brings another issue of antimicrobial resistance being spread across the food chain. So if you're feeding animals with antibiotics, that could increase the selection of some pathogens, antibiotic resistance in these organisms, in these animals, and this can be spread to people that have contact with the animals, but also through the products that they generate, especially meat, the manure that's produced, that ends up being spread on crops, and things like that. So this is like a cycle that we need to be aware and be concerned about, okay? And also there are studies showing that some pathogens that acquire these resistance genes, and this is one of the major issues that's discussion between animal production systems and what happens in the clinical practice, that some of these pathogens can jump from animals to humans. We have been discussing this during the pandemic with the COVID and other diseases that happens with mediated by virus and also by bacteria, but some studies have trying to demonstrate this. This is not an easy thing to demonstrate. See, this is one of the few studies that actually went after this using whole genome sequencing of bacterial pathogens that had multi-drug resistance genes in animals. And they monitored, they isolated zooxanthellae from animals and also from humans. as you can see here. And you see that sometimes you obtain organisms from humans that actually match with the isolates that are obtained from animals. And usually this is more frequently associated the jump from livestock to humans than humans to livestock. So that's like where based most of this discussion about these jumps on production. So in our case, we're more concerned about bovine mastitis. And why we're concerned about bovine mastitis and how this ties to the whole story. First of all, bovine mastitis is the most common disease of dairy cattle worldwide in both conventional and organic farms. It's also the disease that costs the most to the farmer. The estimated cost for clinical case in the first 30 days in milk is over $400 to the producer. The overall losses to the US dairy industry are estimated in more than $2 billion per year. And also, the main reason why this ties to this story is the fact that bovine mastitis is the most frequent reason for antibiotic use in dairy cattle. In Wisconsin farms, 80% of all antimicrobial drugs used at the farm level are used for controlling bovine mastitis. So that's the main reason why these antibiotics are typically used in these animals. And this is this retrospective study that was done recently shows exactly like bovine mastitis is the main infectious disease that happens at the herd level. This was done in 37 Wisconsin farms, covering a large group of animals. And this is just the incidence rate of the disease among these herds. And you see that about 25% of the cows per herd per year are affected by bovine mastitis. So this kind of like ties with the idea that we're talking about. And if you think about what kind of antibiotics are actually used to treat these animals when they have mastitis. So we see a whole bunch of different cephalosporins and also beta-lactams are used to treat, but by far, ceftriaxone has been the active ingredient or the main antibiotic used at the farm level to treat the disease. So keep this in mind because it's going to tie with another thing that I'm going to mention like in a couple of minutes. So one of the things that we're asking in our research group is, can we find alternatives to the use of antibiotics to control this infectious disease in the animals? And if you aim to find something different, where can you go to find these new molecules or new bioactive molecules? A lot of people have turned their attention to soil microbes because they traditionally are capable, especially if you look at the genus Streptomyces, they can produce a variety of compounds that can be used as antibiotics. And if you go to a pharmacy, that's probably something that you're going to buy that comes from these organisms. Okay? But we think that the gastrointestinal tract of livestock are a potential source of novel bioactive molecules as well. And one of the reasons for that is, this is also that really fascinates me. If we look at the composition of the gut microbiome in these animals, and this is a representation of co-occurrence network of rumen microbes in the gastrointestinal tract of cattle. And this is basically networking showing interactions between rumen microbes in the foregut of the animal, and this in the small intestine and the lower part of the gastrointestinal tract. So there's a lot of interactions between these organisms. And if you think about the basics of ecological interactions between microbes in any ecosystem, you see that's really relevant that for their interactions, they need to interact by chemical communication, but also by production of some metabolites. And you can just take a look under the microscope at the feed particles that are colonizing this ecosystem. You see how this actually plays out. Everything that you see here are basically bacteria attached to a feed particle in the room. So they really colonize the environment where they are in. And this is really a great source to find these new molecules, because in order to colonize these niches, these ecological niches, they compete with each other for substrate and they produce molecules. And if you look at the genomes of these organisms, they can produce a variety of different molecules. Some of these molecules will be signaling molecules that they use to talk to each other. Some of them will be molecules that talk to the host, and they influence the immune system of the host in the gastrointestinal tract. And some of them will be directed towards like pathogens or invading organisms that share the same ecological niche. So we can look at the genomes of these organisms and try to identify some of these molecules. And this is something that we're particularly interested. So we direct our attention to this particular group of molecules. The antimicrobial peptides are produced by these bacteria. And the reason for that is because they are considered a new generation of therapeutics. That's a class of molecules that is distinct from antibiotics. They show a great diversity of sequences and secondary structures. They have great stability to extremes of pH and temperature, which is good for practical use. They show different modes of action, low toxicity against mammalian cells, and fast killing and low rate of acquired resistance. There's also a difference that you see compared to other kinds of antimicrobials. And the fact that they're gene encoded, which means that they have a specific gene that encodes these molecules, allows us to modify this gene sequence. We're talking about gene editing this morning. We can actually add the composition of these genes and also improve the spectrum of activity or improve the overall activity and solubility of these molecules as well. And one thing that we were interested is about the synergies that they can present with antibiotics as well. So this slide here is going to resume a lot of different work that was done. Just to put some context here, in order to look for this information in some of these bacteria, we look at the genomes of some rumen bacteria, looking for specific biosynthetic gene clusters that were associated with the production of these antimicrobials, and particularly with the antimicrobial peptides. And we went after a group of bacteria of the rumen ecosystem, an aerobic group of microbes in this ecosystem. There were the streptococci, because they had a complete biosynthetic gene clusters in their genomes. We isolate some of these microbes using phenotype mining of these organisms, and some of them were able, I mean, like Jimena was saying, a picture tells more than like 1,000 words. You see like the capacity that they have to produce antimicrobials that are target-specific organisms. And this morning we're also listening about Listeria monocytogenes. This is exactly Listeria monocytogenes that they were acting against, which is a known foodborne pathogen. But they also act against other organisms, including from the rumen. So this molecule was purified. We did mass spectrometry to characterize the molecule. It is a small molecule that contains around 23 amino acids, has a mass of 2,500 Daltons, and is encoded by a specific gene that's called BVCA. So this is located in the genome of the organism that contains these biosynthetic gene clusters. This is a post-translational modified molecule, which means that after being translated, there are modification enzymes, particularly these two here, that are involved in the modification of the molecule and produce these lengthionine rings that are important for the activity of this molecule as well. So the bacteria also has immunity genes and also regulation genes that are involved in the protection of the producer organism against this molecule. So as we progress with the study of this molecule, we wanted to understand how it actually acts against the target organisms. And again, summarizing a lot of different experiments in a slide, showing how it actually acts against the target bacteria that has activity against. So this molecule that we call bovine HC5, because it was produced by a strain of Streptococcus bovis, actually is capable of binding to lipid 2. For those who are not familiar with the biosynthetic pathway of cell enveloping bacteria, lipid 2 is a crucial precursor of the cell wall in bacteria. So it actually binds to this precursor in a stoichiometry of 2 to 1, and is capable of producing this spore there in the cell wall that allows the leakage of intracellular ions. And bacteria are really loaded with potassium, and once you punch a hole in the cell membrane, they basically cause an extrusion of these ions, and some cases they will lose also ATP, and that pretty much the cell loses viability when they do that. So this was quite effective in acting like that. And the question that we wanted to ask after this is, can we use these molecules to actually target pathogens involved in infectious disease in cattle? And the disease that we were targeting was mastitis. So the initial experiments that we were interested in doing is to use not a single strain of bacteria associated with mastitis, but we wanted to use a mixed culture of streptococci and staphylococcus strains, staphylococci, together. So in this figure here, you see the growth of a mixed culture of 10 different strains of staphylococcus and 10 different strains of streptococcus associated with mastitis. And this is the control without the just like immediate without the addition of any inhibitor. And these are different concentrations of the inhibitor that we wanted to test, which is the bovine HC5 compound. And you see these are different concentrations in two different conditions, in aerobic conditions and under anaerobic conditions. Okay, thank you. So remember that I was mentioning about the use of ceftriaxone as an antibiotic that's used for mastitis control. The next question we had, can we use these molecules in a synergistic way to reduce the amount of antibiotics to treat these pathogens or to control mastitis? So we did a study where we were measuring the feces, which is a combination of this peptide with the antibiotic, in this case, ceftriaxone. This is just a way that you calculate these interactions to determine if the interaction is synergistic or not. And this fixed value represents that. So we use like 19 different strains of Staphylococcus, which is one of the pathogens associated with contagious mastitis. And anything that's below, if you have like an index of 1, this indicates that you have like an additive effect. So half like of the activity will come from the antibiotic, half will come from the molecule you're testing to. If anything is below 0.8, which is drawn here, it's considered to be a synergistic effect. And anything below even that, like even more synergistic. So you see that the average that we got here was 0.653, which is considered highly synergistic in this interaction. And this is also representing this graph that we call isobologram, where we change the concentration of our antimicrobial peptide, and this is the concentration of the antibiotic. So this is the additive level, this is the synergistic level, the threshold, and this is the 0.5 threshold. So everything is in that range, which indicates that the effect is actually synergistic. We also did the interactions with different concentration of these two molecules against a range of these pathogens. And again, here on the x-axis, you see the different concentrations or increasing concentrations of the antimicrobial peptide that we were testing, with different combinations of the antibiotic that range from here to this higher concentration here in the bottom. So the darker the color that you see in this heat map indicates a higher concentration of the antibiotic is needed to inhibit that pathogen, okay? And you clearly see a column effect. As you increase the concentration of the antibiotic, you see that there is a decreased amount, and I know that you guys will not be able to see the values here, but if you compare the concentration of antibiotics that you have here to these columns here, there is a reduction in about 80 to 90% of the antibiotic needed to cause the inhibition of the pathogen, so which is quite good. And this is also evident here. If you look at the minimum bactericidal concentration of these interactions between the antibiotic and the AMP, where over time you see how the column effect plays again. You see the increasing concentration of the antimicrobial peptide that we're testing, and here the increasing concentration of the antibiotics. Because as we increase the concentration of the peptide, you can see the killing effect against the cultures. So it's quite striking the synergism. And it's also demonstrated here with the time kill assay, to where you have the control. Okay, almost done. The control going without the addition of the peptide, and with the addition of the peptide plus the antibiotic. So this is just the antibiotic alone, and this is the combination that we had. And remember, the combination has reduced amount of both the antibiotic and also the peptide together, and they do a very interesting effect here. So we got a reduction that is usually above 10,000-fold in terms of reduction of the microbial population. And the last thing that I want to show is about the study that was done with biofilm disruption. Most of the several mastitis pathogens, several strains of mastitis pathogens, they can actually form biofilm in the udder of the cow. And this is one part of the pathogenesis, how they colonize the microsystem. We knew that these molecules could prevent biofilm formation, but we want to know if they could disrupt biofilm formation once the biofilm was already established. And this is the growth, like the negative control, like before the incubation, after the incubation. So you see like the formation of biofilm will increase after time. And this is with the treatment, with the combination with the peptides, antibiotics separate, and with the combination with the reduced dose of the antibiotic and the peptide, which means that they can really reduce the concentration of these molecules. So another interesting fact that we found is that this antibiotic and peptide combination can reduce the susceptibility of multi-drug resistant strains to the level comparable to the ones that are susceptible actually to to antibiotics, not the resistant ones. And this is demonstrated also for the efficacy values in this slide here. So to finish up, there's a lot of other things that we did on this thing, focusing on the development of antibiotic-free formulation. I don't have time to tell you guys about that. And we also did a pilot study in vivo in using some of these peptides to evaluate the effect on mastitis in vivo settings, injecting these molecules. But this, I will leave for you guys to look at the poster 5 this afternoon because we have more information there. So I don't have time to talk about this today. But it's a very interesting analysis of this not combination of antibiotics and peptides, but just the molecules by themselves. So just to finish up, so MPs are not magic bullets, but could represent an effective and safe alternative to traditional antibiotics, mastitis prevention and treatment. The combinatory therapies are probably a more realistic solution to current therapies. The gastrointestinal microbiome livestock is still a very underexplored resource for antimicrobial discovery, and work is needed to expand our knowledge about the mechanisms of action and greatest synergism against bacterial pathogens. And selection of resistant strains must be evaluated. Thank you.
All right.
Thank you.
Thank you, Larry.
Reduce my presentation.
You know, I just wanted you to clarify something for the audience that aren't familiar with dairy production systems.
Yes.
Cows that are treated with antibiotics on farms, that milk is not saleable.
No, it's not.
You're not going to go to the store and drink milk with
not.
antibiotics.
Yes,
I just want to
this is another
clarify
thing
that.
that is being tested in this pilot study. We're trying to reduce the time.
Okay, questions for Hilario. We got a few minutes here. Any questions for Hilario? Okay, here we go. Carolina.
So you were saying, very interesting, lots of information.
Yeah.
I might need a couple of days to process.
I know. Sorry.
But the question is, you said at the end, like it could be for treatment or prevention of mastitis.
Correct.
So like one of the future studies could be like for dry cow.
Yes, excellent question. Yeah, this pilot study was done with the intramammary infusion of these antibiotic-free formulations. The objective there was like to treat animals that had like high somatic cell count. We used a pilot with 26 cows that were treated with this in different quarters. They had like a total of 48 quarters that were treated, half with antibiotic, half with this antibiotic-free formulation. But one alternative approach for using these molecules that I think would be quite interesting, it would be to do the dry cow therapy and do selective dry cow therapy as well. Not as an intramammary infusion, but like a teat seal or something like that. Not to cure, like a prevention of the infection during the dry cow period.
Okay, other questions? Hilario, I have a question.
Yes.
Is this mechanism that you're talking about, is that specific for types of bacteria? I mean, gram-negative, gram-positive. Is it going to work on any kind of bacteria that it's exposed to, or is it highly specific?
Excellent. That's why you should go to the poster number 5 this afternoon. We are, this molecule that I was particularly talking about is more active against gram-negative organisms. But in the follow-up study that we did, we select some molecules that are also from the rumen environment that have a broader activity against gram-positive and gram-negative. And we use like environmental pathogens and also the gram-positive ones, the contagious ones. And this was in collaboration with Wisconsin Veterinary Diagnostic Lab. They provide us cultures that they collect from farms in Wisconsin, and we use that to assess.
And what about good bacteria in the rumen?
Yeah.
If you're going to treat a cow, I mean, is there going to be any danger of wiping out the rumen microbiota or anything?
No, we are just injecting.
Just injecting.
In the udder, not like
in the udder, but it's not going to go to the rumen.
No, not going to the rumen.
Good. We wouldn't want that to happen.
Yeah.
All right. Good. Let's give Hilario another round of applause. We'll go ahead and get you guys switched.
Oops.
So moving along now, we're going to move into our short talks. And so our first short talk is by Hendyel Pacheco. She's a grad student in animal and dairy sciences at UW-Madison, mentored by Dr. Francisco Peña-Garicano in the animal and dairy sciences department. And she has a BS in veterinary medicine from Brazil. So you're going to be talking about, do we have it up here?
Yeah, just hit next.
Okay.
Yeah.
Okay.
One more.
One more. There we go. The impact of homozygosity on dairy bull fertility.
Yeah. Hello, everyone. So thank you, Paul, for the introduction. Yeah, so I'm Hendyel Pacheco. I'm a PhD candidate in Dr. Francisco Peña-Garicano's lab. And during my PhD, I've been working on bull fertility in Italian brown Swiss cattle. And in the past few months, we had the question if homozygosity can affect bull fertility, or if inbreeding can affect male fertility. So today, in this presentation, I'll give a summary of our findings, and I hope that at the end of this presentation, I can give you the answer. So we know that one of the major challenges in the dairy industry is to achieve rapid genetic progress while maintaining adequate genetic diversity. And we also know that the intense selection for high-producing dairy cattle results in the widespread use of related animals for parents to the next generation. So this selection practice leads to a decrease in the effective population size, a decrease in the genetic diversity, and an increase in inbreeding levels. And one of the major concerns is that this increase in inbreeding levels have a negative impact on the overall performance of the animals in their cattle. And this is known as inbreeding depression. So, and this increase in inbreeding levels also tends to increase the proportion of homozygous loci. And this increase in the proportion of homozygous loci leads to a decrease in fitness-related traits such as male fertility. So you might be wondering, how can we quantify this inbreeding? So inbreeding can be quantified using different approaches. So we can use pedigree information, but we know that usually the pedigree information is not very accurate. So nowadays we can use genomic information. So one of the approaches to quantify this homozygosity is using genomic information. It's called runoff homozygosity. So runoff homozygosity is defined as a continuous stretch of homozygous genotype. And here in this image, we have, in a very simplified way, how is the formation of this runoff homozygosity. So here we have individual A, which represents the common ancestor of parents D and E of individual F. And here in the individual F, we have a fragment highlighted that represents this runoff homozygosity. And this fragment here, this segment, comes from the common ancestor, the individual A. So here, just to illustrate the runoff homozygosity. And then with that, we can measure the length of this homozygous segment, and we can also count how many homozygous segments each individual has. And then we can understand what is the impact of homozygosity on male fertility. So here our objective was to evaluate the presence of homozygosity in the Italian brown Swiss cattle, and then assess its association with bull fertility. So for the analysis, we had 1,100 brown Swiss bulls with both phenotypic and genotypic data. For the phenotypic data, we use circumception rate, or SCR, which is a measure of bull fertility. So the circumception rate in this population ranged from -22 to almost 10%. And for each animal, we had over 450,000 SNP markers. And for the analysis, we used the software called Plink, and we had basically 3 steps. So first, we performed the discovery and the characterization of these runs of homozygosity. We then performed a comparison of this homozygosity between 2 groups, one low fertility bulls and another one high fertility bulls. And we then performed an enrichment of these homozygous regions in the low fertility bulls. So basically, we identified these homozygous regions. We mapped them to the bovine genome to try to identify genes that are associated with male fertility. So now I'll start showing what are our findings here. So we performed the association between homozygosity and bull fertility in 2 different ways. So first, we use the total homozygosity length for each individual versus circumception rate. So here each dot represents one individual, so we have over 1,000 dots. And this line here represents the regression line for circumception rate on the total homozygosity. And as we can see, there is a negative association between bull fertility and the total homozygosity, which means that animals that have longer homozygous regions have lower fertility. And then we perform this same association, but now using the total number of homozygous segments we counted for each individual. And here we also see this negative association. So, which means that individuals with more homozygous regions have lower fertility. And for our second analysis, we performed this association of homozygosity comparing two groups. So we have 100 bulls in the low fertility groups and 100 in the high fertility groups. And here in these box plots, we compare the homozygosity between these two groups. So we can see that the levels of homozygosity between these two groups are different. And the total homozygosity in the low fertility group was significantly higher, which means that low fertility bulls have a higher inbreeding, consequently a lower genetic diversity. And third, we performed the enrichment of homozygous regions in the low fertility bulls. So here we have a Manhattan plot, because we like Manhattan plots. So each dot here represents one homozygous region that was enriched in these low fertility groups. In the x-axis, we have the chromosome number that goes from 1 to 29, so we use the autosome. And here in the y-axis, we transform the p-value to minus log 10. So higher the peak, higher the significance. And with this analysis, our objective was to understand how specific homozygous regions can impact male fertility, and then identify the genes that are within each region. So in this case, we have 4 homozygous regions that were significantly overrepresented in the low fertility group. We have one in chromosome 6, 10, 11, and 24. And it's important also to note that these 4 genomic regions were validated in the entire population. And very interesting is that all these regions harbor genes that are related to, in general, to sperm biology and male fertility. And also something that was very interesting for me is that this region on chromosome 6 was previously reported as a region associated with bull fertility in brown Swiss cattle. And all these 4 genes here, they show medium to high levels of expression in the testes of brown Swiss bulls. So it seems to be a very important region when investigating bull fertility in brown Swiss cattle. So here our findings show that inbreeding and increased homozygosity have a negative impact on male fertility in brown Swiss cattle. And the quantification of runs of homozygosity is a powerful tool because we can understand the population history, we can assess the genomic inbreeding, and we can identify genes that are linked to important economical traits. So the quantification of homozygosity and the identification of those homozygous regions have the potential to help in mating programs when we are aiming to reduce these inbreeding levels, and also avoid the production of homozygous offspring, and also keep the genetic diversity. So I hope I answered the initial question, and if you have some question, I'll be glad to take, and I'll be also in the poster number 7.
All right, great. Let's give Angel a hand. We have time for just a couple quick questions. I'll start with one.
Okay.
Do we know physiologically how inbreeding manifests that decrease in fertility? Is it like a sperm problem with fertilization, or is it like an embryo problem with embryo development? Do we know that?
Yeah, so the genes that I found associated was mainly in sperm biology, but also it can be after the fertilization process.
So
So,
an
it can be embryo development
problem.
too.
Okay,
Yeah.
thanks. Other questions from anybody? Why brown Swiss?
Yeah, everybody has the same question.
Yeah.
We have a good relationship with the Italian Breeders Association for brown Swiss, so yeah.
And plus,
so
it's
then-
fun. It's fun to go to Italy, right?
Yeah, I wish
Yeah.
I could. Yeah.
Okay. All right. Let's go ahead and give Angel another round of applause, and we'll go ahead and get switched over. And I'm going to turn the mic over to Jimena.
Thank you, Paul. Our next speaker is Ligia Cavani, and she's a postdoc in the Animal and Dairy Sciences Department with Ken Weigel and Francisco Peña-Garicano. She received her PhD in Animal Sciences from São Paulo University in Brazil, so please welcome Ligia.
Hi everyone, thank you Jimena for the introduction. The title of my presentation is Consistent of Dry Matter Intake, and we're gonna see if you can actually correlate it with resilience in dairy cattle. So I would like to start in showing daily dry matter intake of 2 cows at the same stage during a trial period. So it's easy to see that the blue cow presented more fluctuation in her daily dry matter intake, right, compared to the red one. It's fair to say that the red cow is more consistent in her dry matter intake daily compared to the blue one. The reason for that, some behaviors of a lot of fluctuation or variation in daily dry matter intake can be correlated or related with stress, right? Or perturbations, you can call perturbations. And the list is huge, can be disease, social interactions, temperature changes, environmental conditions, also some alterations in management and also in diet. And I can keep in saying the list, right? But also there are some that we don't know, and some we cannot record or we cannot measure. Well, but the important thing, really important thing here is the response of the cows face a perturbation are not the same. They're different. And how consistent of the matter intake can be related with resilience? So there is a general concept of resilience that can be defined as the capacity to maintain performance or bouncing back to normal function after a disturbance or a perturbation. So high frequency data, such as daily dry matter intake, actually can tell more about resilience, or at least try to identify those cows. We have a lot of questions about this topic. It's really new, so we just started. But in this study, we want to answer these questions. So first, how to evaluate dry matter intake consistent? And because I'm geneticist, I want to know if it's heritable or not. And also, if is there a relationship between dry matter intake consistent and milk yield consistent? Is dry matter intake consistent genetically correlated with feed deficiency? And how about other important traits? We have a lot, right? And finally, could dry matter intake consistent be an indicator of resilience, the general resilience that I explained? Okay. So we use data from 5 research stations here at UW, Michigan, Iowa, University of Florida, and USDA. So those stations provide dairy cow feed efficient. So then we have daily feed intake, obtained from different systems in Setec Gates that we have here at Arlington, Tystall, and Callan Gates. We have also daily milk production and more milk composition and body weight. In this study, we used more than 6,000 lactating cows, 400,000 daily dry matter intake records, and the period that we used to analyze these traits was around 64 days on average per cow. Consistent of dry matter intake is not a straight for phenotype or trait. It's not easy also to get. So in this study, we have actually 4 steps that we analyze. So first step, just to get the phenotypes. It's first we estimate the expected daily dry matter intake for each cow in this period, 6 days on average. And then we calculate the deviation or the difference between what we expect and what we observed for each cow again, and per day, right? And then the third, we calculated the variance of those difference, of those deviations. And finally, we just transform as log because of the model, the assumptions of the model. Okay, the challenge part for these steps is actually the step 1. Why? How we can actually estimate the expected daily dry matter intake in this period, 6 days. It's not, we don't have an answer yet, so then we try 5 models. So we start with the simple one, that's just the average, the cow. And then we try the quantile regression, use the quadratic and cubic. And we also try low-S regression, that's no parametric model, that we can model no linear trends. So this is an example of a cow that's pretty consistent. So just to illustrate, this is another cow that's pretty the opposite, inconsistent. Just to see how the models fit. So this again is to get the expected value of dry matter intake. So then we just calculate the deviation and then the variation. So the first table that I want to share is the heritability of the trait. So then we have the trait for consistent of dry matter intake is the variance of daily dry matter, the deviation of dry matter intake, and then using different models. And then we got a heritability range from 0.11 to 0.14. Who is quantitative genetics, who work with quantitative genetics, this is moderate, is not low heritability. So this is an indication that we can improve or we can have more consistent cows related to dry matter intake through genetic selection. This is good. So answer that question, yes, it's heritable. Okay, but like I said, we can calculate different ways using just models that we can calculate the expected value. So we use 5 models, but it's matter, the model or not, at least in our data set. So for that, we calculate genetic correlation and also the correlations of sires PTAs, because we want to see in a genetic point of view. We have quite strong and positive correlation, close to 1, so actually it doesn't matter much which model we used. Again, we have a short period, right? Maybe a long, it matters. Other question, there is some relationship or association with consistent of dry matter intake and milk. So to answer this question, we calculate the consistent of milk as exactly the same way that we did for dry matter intake, using the same period, using the same models, and then we got positive and strong genetic correlation range from 0.51 to 0.62. So it means that selecting cows more consistent of dry matter intake, daily dry matter intake, we also favor cows more consistent on daily milk yield. It's a favorable correlation. This is one of the most important questions that you want to answer, that is the relationship between consistent of dry matter intake and feed efficiency. So for that, we again, perform genetic correlations and experiment run correlation of sires PTAs with milk energy, metabolic body weight, and residual feed intake. In general, we got positive and strong correlation, some moderate correlations here with residual feed intake, but this is an indication that cows that are less consistent of dry matter intake, or has more cows that have more variation, those cows are less efficient. Finally, how about the relationship between dry matter intake consistent and other traits in the US national evaluation? Those traits can be grouped as production, longevity, health, repo, and feed efficiency traits. This table is the experiment correlation of sires PTAs. So I'll just point some things here. So here we found the unfavorable correlation with milk yield. In general, no important correlations with longevity and health traits. Interesting that we found a favorable correlation with heifer conception rate, and reinforce the results that I showed before. Again, the cows that are more consistent or less variation are more efficient. As take-home message, so dry matter consistent traits are heritable. They are genetically correlated with milk yield consistent in a favorable way. They're also in a favorable way genetically correlated with feed efficiency trait. And finally, we can say that dry matter intake consistent could be an indicator of resilience. Why? Well, cows with lower variation in their expect daily dry matter intake may be indicated their capacity to maintain performance in the face of challenge, stress, or perturbations. And we also saw that cows with greater variation means less consistent in the way that we show the trait, are less efficient and may be less resilient. So with that, I would like to thank you for your attention, and then I also would like to thank the Dairy Innovation Hub, USDA NIFA, FFR, CDCB for the support, also the Feed Efficiency Group here at UW, and Francisco and Kent's lab, my colleagues. Thank you.
All right. Time for one question. No questions. I have a question.
Mm-hmm.
Do you know the cause of the inconsistency for the individual cows in your study? I mean, it could be behavioral, it could be lameness, it could be sickness, it could be a lot of different things. Do you know what was associated with the variability?
This is the next step.
Okay, that's the next step.
So the next step is actually to try to look the data the other way. So then suppose that I have a huge probability in that way something happened, and then see the difference of the cows.
Good.
Why some cows doesn't have any impact or some has.
Okay, great. Thank you. Let's go ahead and get switched over. Our next speaker is Guillermo Martinez Boggio. He's a postdoc in animal and dairy sciences, mentored by Kent Weigel and Francisco Pena-Garicano. Some popular names here this afternoon. He received his PhD in animal science from INRAE in France. And go ahead and hit the button here. He's going to talk about host rumen microbiome interplay affects feed efficiency in dairy cows. Go ahead.
Hi, everyone. Thank you very much, Paul, for the introduction. So, as Paul said, today I will talk about how the interplay between the host and the rumen microbiome affects the feed efficiency in dairy cows. So I was thinking in the picture that Hilario presented in his presentation, and he showed this interaction that have the microbiome and also in the rumen, but also this microbial community have an interaction with the host. So then we can consider that the animals are no longer individuals, they are holobiont. Because the host and the microbiome form a complex and interconnected ecosystem. So I try to represent this ecosystem through this diagram, in which we have three components, the genome, the microbiome, and the phenotype. So the single arrows shows the effect of the genome and the microbiome on the phenotype, and the double arrow, the interaction between them. So our objective was to quantify these effects and also the interaction, and to evaluate the improvement that we have in the prediction abilities for the feed efficiency traits. So our data set consists of around 448 multiparous Holstein cows, and using the previous diagram, in our work, we represent the whole genome using the genotypes, 79,000 SNPs, the microbiome, and here we use the rumen microbial community, because as you know, in the ruminants, the microbial community in the rumen gives the ruminant the ability to transform plants in high quality products. So this rumen microbial community we represent with the abundances. Then we use the 16S RNA abundant gene, and we amplify in a specific region that is the B4 to have information about bacteria. So we have in our dataset around 3,000 amplicon sequence variants, but for this presentation we call just bacteria. And finally, the phenotypes. And here we use the feed efficiency trait, residual feed intake, dry matter intake, and net energy secreted in milk. So how we integrate this component in the molds? So we use a multi-omics approach, and here we use three different molds. The first mold is the mold G, in which the phenotype is explained by the genetic effect. And through this mold, we can estimate the heritability, the proportion of the phenotypic variance that is explained by the genetic effect. But then, to this mold, we add the microbiome effect. So here, in this mold, what we can capture is the direct genetic effect on the phenotype, the microbiome effect on the phenotype, but here, with this, in this way we can capture the indirect genetic effect through the rumen microbiome. And here we model through this model GM, and for the people that doesn't like quantitative genetics and you learn just heritability, so here we have the direct heritability and also the microbiability. That is a term defined in 2018, in which simply is the proportion of the phenotypic variance explained by the microbiome. But then we use another mold, in which for this mold GM we add the interaction. So here we can call the Olovion mold or mold GMO, that is nothing about the GMO that Alice presented this morning. It's nothing about genetically modified organisms. It's just a mold, and I call it this way, was my, my way. So then, through this mold, and again, here we can estimate the holoviability. It's the proportion of the phenotypic variance explained by all these effects. But then here you are thinking how this guy will integrate all these high-dimensional omics in a single regression model. And the answer is kernels. So I use multi-kernels mold, regression models, that is a very elegant way to include this high-dimensional data in the regression models. So let's see what I obtained. So what is the contribution of each omics on the phenotype? So in this plot we have 3 segments. Each of them represent one different trait: residual feed intake, dry matter intake, and net energy secreted in milk. And the boxes represent, here we have the heritability, and as you can see, the heritability is larger than the direct heritability for all traits. So that shows that we have the microbe mediate part of the host genetic effect. So with this model, in which we include the genome and the microbe, we can capture this indirect genetic effect. Also, as you can see, the microbiability was a moderate microbiability. So we have a moderate contribution of the rumen microbiome on the phenotype, on the feed efficiency. And finally, the alloviability, the gray box, as you can see, for all traits was larger than the individual components. So we are captured, or we can quantify this interaction between the genome and the microbiome. So then the natural next step was to see if this contribution of each of these effects improved the prediction ability for the feed efficiency. And here we use a cross-validation, a tenfold cross-validation. Here are the results for the Spearman correlation and the mean square error for the prediction in this figure. Again, each segment represents one trait. And as you can see, the green and the gray box are the moles. In green, the mole GM, and in gray, the mole GMO. So this mole, when we include the rumen microbiome, we improve the predictions for the feed efficiency traits. But here, the rumen microbiome is included in the mole as the whole microbial community. We are using all the information that we have. So the idea at this point was to try to identify which bacteria or which genera are impacting more in the feed efficiency traits. So to do that, we took this mole GMO, the Olovian mole, and when we include the microbiome and the interaction between the microbiome and the genome, we compute 44 different matrices, and in each matrix we include one different genus. So the bacteria that compose the microbial community will regroup in their genus level, and we use one at a time in this matrix. So as you can see in this plot, each line represents one of the traits, the y-axis represents the alloviability, and here we have all the genus that we have. So as you can see, we have some of them that contribute more than others, the same here, for the phenotype, on the phenotype. So some of them, and I will not take the risk to pronounce them because I will make a mistake, you can ask Hilario after how to pronounce this, contribute more for some of the traits. So then again, the natural next step was to include these genus in the prediction models. So here I present the model with the genetic effect, the Olovian model with the 3 effects, and all the other boxes are the models when we include each genus at a time. So as you can see, as a general pattern, when we include each genus at a time, we improve the prediction with the model that include only the genetic effect, but only some of them, mainly what I present before, these genus that I present before, improve the prediction with the Olovian model. But of course, maybe for the small data set that we have, we need to test this result in a larger data set. So, as a take-home messages, the rumen microbiome has a moderate contribution to the feed efficiency traits. The Olovian model, this model GMO, yields better phenotypic predictions. It's just an effect of the presentation to...
Everybody, please silence your devices.
And the last point is the microbiome mediates part of the host genetic effect on the feed efficiency trait. So the next step of my work is to go deeper and try to, once we identify this indirect path of the genomic on the phenotype, try to incorporate to estimate the PTAs or the breeding values of the animals. Thank you very much for your attention, and I'm happy to take your questions.
I told my wife I would be moderating a session right now, so, okay, we got a question right here.
Yeah, so my question is, Yeah, the question is, so, have you tested if there is a genetic, so if genetically similar cows have genetically, microbiome, similar microbiome diversity, and yeah, go ahead.
Yeah. No, I didn't test, but what I think that what you are saying, if we have some cow that genetically is better, if they have more similarity in terms of microbiome, something about that.
If there is a relation, yeah.
Yeah, no, I didn't test if they have a. Yes,
Groups that have genetically clustered together and they are microbiome similar.
Yeah, I didn't do this kind of discriminant analysis in which I can group the animals based on the genome. We can use maybe a good idea could be use the genomic matrix and the microbiome matrix and try to group the animals based on these matrices. I didn't test that, could be a good idea to do. But the idea was just to incorporate the information, the similarities that we have in terms of microbiome and genomic. But commonly what we have is the microbiome depends a lot the trait that we are using, if they have a contribution or not, or if they have a similarity or not. So.
All right, great. I have a question while we switch mics. So let's get the last speaker up here. We can feed ionophores to cows.
What?
You know what an ionophore is? Like rumensin?
Yes.
It would change the rumen microbiome.
Yes.
What would happen to your predictions if you had cows that were treated and untreated with?
That could be a problem.
That could be a problem. But
Yes,
your model should be able to predict that.
You know, because we will change the composition of the microbiome, the abundances will change.
I think that'd be interesting to look at.
So the similarity will change.
Great. Thank you. Let's give him one more hand.
All right, we made it to our last speaker. Thank you for staying in this great section this afternoon. Now we have Larissa Novo. She's a graduate student in the Animal and Dairy Sciences Department, and she's mentored by Professor Francisco Peñagaricano one more time. She's a master's in the animal science from Federal University of Mato Grosso in Brazil. So please welcome Larissa.
Thank you. Is it on? Yes. Yeah, okay. I cannot hear me. Oh, yeah. I don't know how I can present after Guillermo because he was killing it. Today I'm going to present a feasibility study of genetic evolution for Johne's disease in US Holstein cows. So I'm going to start talking about why are we studying Johne's disease? Because it's not a new disease, it has been here for over a century. We are trying to control for it for a long time. We had government program that was trying to help producers and as an incentive to make them control it. But we have a lot of problems doing that. So one of the reasons are here. So this disease is caused by a bacteria, and that has a long incubation period. So the animal will be infected, mainly as a calf, and it will take a long time to detect that. So while we are not detecting, the disease is spreading in the herd. We have a lot of important inconsistencies for testing methods. So we can test via fecal samples, milk samples, blood samples, PCR, ELISA, and even with the same type of tests, we have different protocols that are being used, so it's complicated to standardize that. We do have a vaccine for that, but it has a very strict criteria for vaccination. For example, in the United States, only replacement heifers and bull calves between 1 and 35 days of age are currently eligible to receive the Johne's disease vaccine. And even if you decide to vaccinate your animals in your herd, the animals that will be vaccinated will have more problem to detect the disease because usually the test that is used in the herds is the ELISA test score. So it will be a problem if you're trying to control for that. So we were thinking, how can we try to control for the disease in a way that is kind of independent of those factors presented here, that will not depend so much of protocols or the decisions that the producer make. And we thought about a feasibility study. So on top of those problems that I presented before, here is an example of the data that we do have. It comes from CDCB, from the Council on Dairy Cattle Breeding. And here is an example that shows different protocols. So before 2011, almost 100 of the tests were from one protocol. And after we switched, so this makes a problem for us trying to use both at the same time in a unique genetic evaluation. And we have a problem also with the distribution of the tests over the country. So we have tests from all over the country, but they are very specified for some states, and mainly they are from Minnesota, Wisconsin, and Michigan. So we think that working with a feasibility study may help to reduce the incidence of the disease. So for this work, we are working only with Holstein cows, and we have 365 cows with one information each. These are the states that we have data from, again, mainly from Michigan, Minnesota, and Wisconsin. And we decided to treat the phenotype, the trait, as binary trait. Either the animal is with the disease, with a positive result, or a negative result. And we decided then to separate the protocols. We could not work with them together, so we had 271,000 with IDEX test and 94,000 with Paracheck test. We then used different models to test how the trait and phenotype would behave. We tested either bivariate or a linear model. And we separated the animals by the herd year season, age within lactation, because they varied a lot, and 3 levels of days in milk. Here is the summary of the models that we tested for the genetic evaluation. So first, we tested only with pedigree. Then we added genomic information, and then genomic and pedigree information, but as a linear trait. And okay, we are talking about a genetic evaluation, so what matters in a genetic evaluation? So first, genetics has to be important for the trait. So there's no point of working on that. So we have to evaluate if the trait is editable, and if it is, how much. If it is editable, if we can predict the trait for the animals that we don't have information, or maybe we want to know previously if the animal will have a more chance to get the disease or not. And if we can do that, if we can trust the results that we are getting. With all those 3 informations, then we can have a hint of the work, if it's feasible or not. So here's the first part, if the trait is editable or not. Mainly, it is for the threshold model that we used. For the linear model, we had a lower editability value, but we can say that the trait is editable. Here's the part where we analyze if we can trust the results that we are having. So this is for the IDEX test, the one that we have more information. We saw that adding the genomic information, that it's the second and third row, we can have more animals that shift from lower reliability to higher. So we have more animals in the last category with equal or more than 0.5%, 50% of reliability. So that was a good indication that adding genomic information can help for the predictions and for the genomic evaluation, the genetic evaluation. For Paracheck, we didn't see kind of this pattern. We added genomic information, and we didn't see like that last column getting higher. But we saw that, for example, for the second column in the first row, that we have lower animals in this category, and they are more like spread out in other categories. This is the genetic trend for the disease, and we can see that through the years from 2002 and 2020, we had a negative genetic trend. That means that the animals are getting more resistant or less susceptible for the disease. And we are wondering if we are not selecting for this trait, if we are not including it in our genetic evaluation, how can it be that we have a negative trend if we are not including it? So we were wondering if it's related with other traits that we are selecting for. So this is approximate genetic correlations. So we have what you were expecting. So animals that are more susceptible for the disease, for Johne's disease, they will produce less milk and have higher levels of somatic cell score, shorter productive life. And these are the most interesting results for me. Like we can see a positive correlation for somatic cell score and a negative for productive life. So they will stay less, shorter period in the herd. And all those results, they agree with the phenotypic trend that we are seeing from the last decades. Yes, we are not selecting for the trait yet. Yes, we have a negative trend for the trait. The animals are getting less susceptible for Johne's disease. But one of the questions that I get when I present this work is like, it seems that by the results, we are not doing anything and it's working. So why does it matter work with genetic evaluation for this trait? So we are getting good results, but we still have 75% of the herds in the United States with the disease. The disease still causes around $200 million per year of economic impact. So we are doing something that is working, but clearly is not enough for the industry and the herds in general in the country. So for take-home points, we can say that despite no prior genetic selection for Johne disease, we have a negative and significant trend. Genetic evaluation of Johne's disease susceptibility is feasible in the United States, Holstein cows. And the genetic evaluation has the potential to reduce the Johne's disease incidence rate, mainly in a long term. And that's it. I would like to thank the CDCB for the data, information, and my internship period last year, and my funding sources, CAPES and Fulbright Brazil. And that's it.
Thank you. We have time for a few questions for our speaker. No questions. You guys are a quiet group.
It's
like teaching class on campus.
So, Larissa, what's the outlook for testing going forward? Is the situation likely to get better in terms of data availability and that, or you see what I'm asking, right? Is it,
yeah.
Or are we kind of stuck with the spotty testing?
So in the past, there was a program from the government, USDA. They were helping fund the producers to test for the disease, and now this program ended. So I think that the testing will decrease a little bit. A good point is that they are standardizing all the tests. For example, the first protocol that we had and ended, they shift and they change it the way they were evaluating. Now both of them use the same protocol. But yeah, it may have it may be a problem in the future because we do need phenotypes to keep testing, keep doing genetic evaluation. With genomic information and predictions, we can do it with, like, for animals that don't have phenotype yet. But I think the main key will be the government helping it, and groups that will analyze that, and standardization of the trait. So that may help in the future.
Great. Let's give our speaker one more round of applause.
All right, thank you everyone for staying. On behalf of Paul and I and the Dairy Innovation Hub, we wanted to thank you, everyone, for being here, to our speakers, for staying on time. We did a great job, so now we have some time for a bathroom break. And please meet us at the Alumni Hall in the first floor. We have the afternoon plenary session. There's also posters afterwards, and there's a reception, so keep your tickets and have a drink later. So thank you again. Bye.